Hello everyone. In today's video lecture of neurophysiology, let us discuss regarding propagation and conduction of nerve impulse. So we all know regarding the nervous system. Nervous system is made up of neurons, and these neurons they help in transmission of a stimulus from the peripheral organs or tissues or cells to the central nervous system, so that bringing about a response, a stimulus will be reached to the processing center in the brain, and the response has to be carried to the target cell or target tissue. So in this case, now how exactly this process of transmission of nerve impulse takes place from one neuron to other neuron, we shall discuss. Now, as we all know, a brief introduction regarding the conduction of the nerve impulse. Now, the nervous system is made up of neurons. Neurons are known as the functional unit of the nervous system. Neuron is made up of a cyton and axon. Now, with respect to the neuron, it is known as an electrically excitable cell. Now, what do you mean by that? So, when some ionic conduction happens, now the electricity can pass through the neuronal membrane and bring about excitation of the cell. Due to the ionic difference, a neuron at the rest, it is in polarized state. Outside the neuron, extracellular fluid, it is positively charged, and inside the neuron, it is negatively charged. Because of this potential difference, we say a neuron at rest, it is polarized. Now, whatever the potential difference that we have during the rest of the neuron, that is known as the resting potential. And later on, once the stimulus is generated, at that time there is a formation of the graded potential and the action potential, and this is possible because the neuron is electrically excitable cell. So let us see how all this happens. Now, when we talk about a nerve impulse generated due to stimulus in the neuron, it has to be propagated. It has to be propagated from the region of the nerve impulse generation till the tip. So, if it is generated here, it has to travel through the axon till the end of the neuron and the next neuron that is present here. Okay, so it has to be communicated to the next neuron that is present. So one way, the impulse that is generated has to travel through the axon. This is called axonic conduction. And other way, once it reaches the tip of the neuron, it has to be communicated to the next neuron. So that is known as the synaptic conduction. Okay. So in this lecture today, we shall be discussing how exactly this takes place. Let us move on. Now, first of all, as we have told, a neuron is at rest. It is having some potential, and that is known as resting potential, we told. And that resting potential is around 70 minus 70 millivolts. Now, when there is a stimulus to the neuron, if it is a weaker stimulus or less than a threshold stimulus, it will be generating graded potential. And at the same time, if it is a threshold stimulus, if a threshold stimulus is given to the electrically excitable neuron, then the resting potential will change because of the ionic flow. Okay. Now, because of the threshold stimulus, the potential, resting potential of the neuron is totally reversed. As I have told before, it is positive outside the membrane and negative inside the neuronal membrane that was in polarized state. Now, exactly reversal of charges took place. That means outside it became negative and inside it became positive. So, because of that, we call it as depolarization. So if you consider this as the neuronal membrane, at rest it is positive outside, negative inside. Once there is threshold stimulus, once there is threshold stimulus, it leads to reversal of the charge, that is negative outside and positive inside. How exactly this happens? Whenever there is a stimulus to the neuron, then the threshold stimulus can open the sodium channels. Sodium channels are present here on the membrane. These sodium channels will open up. As a result, sodium ions flow into the axonal oxoplasm. So, okay, so due to the entry of the sodium ions, the oxoplasm becomes positive. At this spot, where exactly the stimulus is given, there is a reversal of the charges. Now, as soon as the electrical depolarization takes place, the depolarization is propagated to the next region. From this region, it has to be propagated to the next region. This will occur by stimulating the sodium channels that are present here. ಅಂದ್ರೆ ಈಗ ಇಲ್ಲಿದ್ದಂತಹ ಸೋಡಿಯಂ ಚಾನಲ್ಸ್ ಅನ್ನ ಸ್ಟಿಮ್ಯುಲೇಟ್ ಮಾಡಿ ಅವು ಓಪನ್ ಆಗಿ ಸೋಡಿಯಂ ಇಲ್ಲಿಗೆ ಬರುತ್ತೆ, ಎಕ್ಸೋಪ್ಲಾಸಮ್ಗೆ ಬರುತ್ತೆ. ಎಕ್ಸೋಪ್ಲಾಸಮ್ಗೆ ಬಂದ ಮೇಲೆ ಇಲ್ಲಿ ನೆಗೆಟಿವ್ ಇದ್ದಂತಹದ್ದು ಪಾಸಿಟಿವ್ ಆಗುತ್ತೆ ಈ ಜಾಗದಲ್ಲಿ. ಓಕೆ, ಸೊ ಈಗ ಸ್ಟಿಮ್ಯುಲೇಟ್ ಆದಂತಹ ಜಾಗ ಇಟ್ ವಿಲ್ ಇನ್ಫ್ಲೂಯೆನ್ಸ್ ದ ಸೋಡಿಯಂ ಚಾನಲ್ ದಟ್ ಇಸ್ ಪ್ರೆಸೆಂಟ್ ನೆಕ್ಸ್ಟ್ ಟು ಇಟ್. ಆ ಪಕ್ಕದಲ್ಲಿದ್ದಂಥ ಸೋಡಿಯಂ ಚಾನಲ್ ಇನ್ಫ್ಲೂಯೆನ್ಸ್ ಆಗೋದ್ರಿಂದ ಇಲ್ಲಿ ಸೋಡಿಯಂ ಅಯಾನ್ಸ್ ಎಂಟ್ರಿ ಆಗಲಿಕ್ಕೆ ಶುರು ಆಗುತ್ತೆ. ಆಸ್ ಎ ರಿಸಲ್ಟ್, ವಾಟ್ ಎವರ್ ದ ಡಿಪೋಲರೈಸೇಷನ್ ಅಕರ್ಡ್ ಹಿಯರ್, ಇಟ್ ವಿಲ್ ಮೂವ್ ಟು ದ ನೆಕ್ಸ್ಟ್ region. ಅರ್ಥ ಆಯ್ತಾ? ಸೊ now what happens as soon as this is happening simultaneously, just half a millisecond after this is happening, the potassium channels will open up. Okay, so in this region, in the region 1, potassium channels open up and they start moving out. So as a result, so you can see here, after half a millisecond, the potassium channels, voltage gated potassium channels open and sodium channels close. So this brings back the resting potential. This brings back the neuron to the resting potential, and that phenomenon, what we call it as repolarization. Okay, so due to the entry of the sodium ions, there is reversal of charges. It led to depolarization. Now the electrical current has moved on to the subsequent region next to the stimulated region. And in the region 1, there is closing of the sodium channels and opening up of the potassium ions. The potassium ions leave. As a result, the um, resting potential is brought back, and the neuron at that region, it becomes again minus 70 millivolts. Okay, so all this happens in a very, very short while, that is 1 millisecond. So sometimes if there is continuous leakage of the potassium ions, as a result, the resting potential, that is a minus 70 millivolts, this keeps on increasing, minus 75, minus 80, like that, because more and more potassium ions are moving out. As a result, the oxoplasm, it is becoming more and more negative. This leads to what is known as hyperpolarization. Okay, because the potassium channels to close down, they will take time. There is slow closing of the potassium channels that may lead to hyperpolarization. Now see, in this diagram, if you see, this membrane is at rest. As a result, there is more potassium ions, organic ions and all. It causes negative potential. And there are more sodium ions here. And because of that, there is positive potential, and this potential difference is what is called as minus 70 millivolts. So that is at the rest. Okay, now what happens when there is a stimulus? When there is a stimulus, you can see, so this is a minus 70 millivolts that I was talking about. This is a neuron is at the rest and it is at the polarized state. This point, the neuron is at the polarized state. Once there is threshold stimulus, once the threshold stimulus comes here, now there is opening up of the sodium channels. As a result, there is reversal of the charges, leading to what is known as the depolarization of the neuron. This depolarization reverses the charges. As a result, you see the positive potential. That is why we call this condition as a depolarized state. Okay. Now, now the action potential is generated by depolarizing. Then, once this is done, um, the current moves to the next region and sodium channels close, potassium channels start leaking. As a result, there is bringing back of the resting potential. This is called as the repolarization. If the leaking of the potassium ions still continues at that time, it leads to what is known as hyperpolarization, which is even more than the negative potential which the neuron has at rest. Okay, so this is what the graph is explaining about. Now, so this action potential that is generated at the point of stimulus, it has all or none principle. What do you mean by all or none principle? If a threshold stimulus is given, the neuron responds by generating the action potential, which is same for all. Okay, the stimulus is equal, or if it is stronger than threshold stimulus, it is always generating action potential which is of 30 millivolts. So that is what is known as the all or none principle. Now, once the action potential is generated, it starts moving. It start moving from one region to other region without change in its amplitude. So the movement of the action potential is without change in its amplitude. Now, so what about the speed of conduction or rate of conduction? The rate of conduction or the speed with which it is conducted depends on number one, myelin shape, presence or absence of the myelin shape. Number two, the diameter of the nerve fiber, and sometimes it also depends on the temperature. These are the factors that can affect the rate of conduction of the nerve impulse. In this picture, you see here, at rest, there is positive potential outside and negative potential inside. There are more sodium ions outside and more potassium ions inside. As a result, there is a resting potential of 70 millivolts. Now, once the neuron is stimulated, now it is stimulated. As a result, in this area, there is reversal of the charges. So this is the polarized state. Now, due to the stimulus, threshold stimulus, now here the process of depolarization are taking place. At this area, you consider this as A area. In this A area, there is depolarization. Why it is taking place? Say, in this area, there is opening up of the sodium channels and sodium ions leak into the oxoplasm. As a result, this area becomes positive and this becomes negative. And once this is done, after half a millisecond of this, the potassium channels open up and potassium ions start moving out of the oxoplasm. Uh, and during this, the sodium channels close, and the current from here moves to the next region. It opens up sodium channels here again. Now the current that is generated here due to the stimulus, it has moved on to the next region. Now sodium channels open here. Simultaneously, potassium channels are leaking here and the sodium channels are closed. Now due to this leakage of potassium channel, it brings back the resting potential here, that is minus 70 millivolts, and here there is depolarization. Okay, so this is how the axonal conduction or the conduction of nerve impulse to the axonal membrane takes place from one region to the other region. Now in this diagram also you can see this is the resting potential where there are more potassium and more sodium outside, and here you can see stimulated when sodium channels open up, sodium enters in, causes depolarization. After of a millisecond, potassium channels start moving out, and current from this area moves to the next area where there is opening up of the sodium channels. Simultaneously, there is closing of this sodium channel, and potassium ions start moving out. Continuous leakage brings about repolarization. Further, if it continues, it brings about what is known as hyperpolarization. Okay, so that is how the axonal conduction occurs. So this picture also explains regarding how the charges move. This area, this is depolarized, and it is moving to the next area. Now, so the in the same axon, there is a phenomenon known as saltatory conduction. So, in the previous slide, we have discussed the rate of the conduction of the nerve impulse depends on whether the neuron is myelinated or not. Now, in case of a myelinated neuron, as we all have seen, there is myelin deposition like this, right? So, these are called as the myelinated neurons. Now, what happens? This myelin that is present acts as an insulator and blocks the flow of the electric current. In non-myelinated neurons, till now as we have discussed, if area A is depolarized, it automatically stimulates the sodium channel next to it, and automatically the current start flowing to the axon. But in case of myelinated neuron, the myelin sheath that is present, this acts as an insulator. As a result, the current cannot move here through the axonal membrane. Now, what happens now? Instead of moving through the axonal membrane, the current start moving through the ECF of the myelin sheath through exoplasm. So it is flowing through ECF of the myelin sheath and exoplasm. As a result, the depolarization which has been brought here in this area, it moves directly to this area. And in structure of the neuron, we know very well this gap between two myelins is what is known as node of Ranvier, correct? So this node of Ranvier gets depolarized. So in this node it is depolarized. This brings about depolarization in the next node of Ranvier, and from this to next node of Ranvier, this is appearing as if the action potential that is generated here, it is jumping from node of Ranvier to node of Ranvier. So that is why this is also known as jumping action potential. So it is very clear here, the flow of current is not continuous, it is jumping from one node to another node. As a result, there is increase in the speed of conduction. So we need to understand myelinated neurons, the speed of conduction is more. So you can see here there is jumping of the action potential. Now, next, this is regarding the axonal conduction in myelinated neuron and axonal conduction in non-myelinated neuron, both we have seen. Now, the next type of conduction, it is from one neuron to other neuron or from one neuron to the target cell. This type of a conduction, we call it as synaptic conduction. So, the physiological connection that occurs between two neurons, what we call it as a synapse, and through this synapse the conduction occurs. Now this conduction is in the form of two ways. Now when we talk about the synapse, there are different types of synapse depending on which parts have been connected. So the next picture explains it better. Now see, this is axodendritic, this is dendrite, this is axon. Then synapse between axon and dendrite, synapse between axon and cyton, synapse between axon and axon, dendrite and axon. So this is the synaptic conduction. These are the types of synapses based on what is connected. Now, what is there in a synapse? In a synapse, we have importantly, so I'll explain this diagram. We have importantly two structures, two membranes. So this membrane of the synaptic knob is known as presynaptic membrane, and this is known as postsynaptic membrane. Okay, and in the presynaptic end bulb, there are vesicles filled with the neurotransmitter. Okay, neurotransmitter filled vesicles are present, and there is rich voltage gated calcium channels that are present. Okay, and the gap between pre and post synaptic membrane, this is known as synaptic cleft, and what is getting released into the synaptic cleft, it is the neurotransmitter. Okay, So this is the structure of the neurotransmitter, and these structures that are present on the postsynaptic membrane, they are receptors of neurotransmitter. The neurotransmitter that is released bind to these receptors. Now there are two important types of synaptic conduction, electrical synapse and chemical synapse. In case of electrical synapse, there is a physical connection between two neuronal cells, and they are connected through what are known as the gap junctions. Gap junctions, as we all know, they are the tubular protein structures that are present connecting the two cellular membranes. And because there is a physical contact, the electrical synapses can conduct very fast, and at the same time, electrical synapses can excite many neurons at a time because of the physical connection. But most of the neurons, they prefer using chemical conduction in case of synaptic transmission. Now, this chemical synapses, they use chemicals known as neurotransmitters. This we shall be discussing in the next chapter of the same unit, neurophysiology. Second chapter is regarding the neurotransmitters. These are the chemical molecules that are produced by the presynaptic cell, and they will help in communicating between the two cells. Next. Now, what happens in a synaptic transmission? Now, as the action potential reaches the neuronal end bulb, it stimulates the voltage-gated calcium channels, and voltage-gated calcium channels open up, and these calcium ions, they fuse to the vesicles, neurotransmitter vesicles, and release the neurotransmitter into the synaptic cleft. Now, once the neurotransmitter is released, the neurotransmitter goes and binds to the respective receptors that are present on the postsynaptic membrane, as we have seen in the previous picture. So the neurotransmitter that is released goes and binds to the receptor that is present on the postsynaptic membrane, and binding of the neurotransmitter either brings about depolarization, or through some secondary messenger, it may bring about depolarization or hyperpolarization in the postsynaptic membrane. Okay, so if it brings about depolarization, it brings about excitation, and if it is hyperpolarization, it brings about inhibition of the postsynaptic membrane. Okay, and once the neurotransmitter function is done, the neurotransmitter will be removed or broken down through suitable enzymes. You can see here, same thing, receptors are present, neurotransmitter getting released, and it will bring about the synaptic action in the postsynaptic membrane. Now, regarding the electrical synapse, as I have told you already, they have tubular protein structures that are present here, and they have a direct contact between the pre and postsynaptic membrane. And because of that, there is faster conduction in case of electrical synapse, and there is group stimulation of the neurons or the muscle fibers due to the physical contact, and there is two-way communication here. It is not the case in case of the chemical synapse. So all this will help you to differentiate between electrical and chemical synapse. So this diagram shows the difference between electrical and the chemical synapse. So you can see here there is a synaptic cleft here, and neurotransmitter release. And through chemical, the information has been conducted to the postsynaptic neuron. But here you see, due to the presence of the gap junctions, there is direct physical contact, and the electrical current can flow through and bring about excitation in the postsynaptic membrane. Then, now the same picture, same details it is showing. So finally, so we in this video lecture, we spoke about how action potential or nerve impulse gets propagated through the axon and between the two neurons, or between a neuron and the target cell. Now, in case of transmission through the axon, two types of transmissions we have seen: transmission in a non-myelinated neuron, transmission in a myelinated neuron. Okay, so and in synaptic transmission, we have seen the transmission through chemical synapse and transmission through electrical synapse. Okay, so that's it. Thank you for watching. These are the references.